Hello students, today we'll be studying RNA polymerase. In the last video we had studied about transcription and we were very much aware of the repetition of word RNA polymerase. RNA polymerase, as was very obvious, is the enzyme, the foremost enzyme for transcription. But now let us study the structure of RNA polymerase and difference between the RNA polymerase in prokaryotic transcription as well as eukaryotic transcription. Fundamentally, it is RNA polymerase is similar in all the three classes: eukaryotic, prokaryotic. But there is, apart from that, there is slight difference. Like for example, bacterial RNA polymerase is composed of two large subunits, beta 1 and beta, two smaller subunits of alpha, and one copy of fifth subunit, omega. This omega is actually not essential for transcription, but it stabilizes the entire enzyme. What does it mean? That beta 1, beta and 2 alpha subunits, they are stabilized because of presence of this omega subunit. It helps in assembly of all the 4 subunits. And eukaryotic RNA polymerase have several additional smaller subunits associated with this core complex. This alpha beta is the core complex. Like in this diagram we see RNA polymerase, it covers approximately 35 base pairs of DNA. And yesterday we had studied that the transcription bubble is of the size of 14 to 15 base pairs. And DNA RNA hybrid is about 8 to 9 base pairs. So this is the schematic model of transcription. Again, I will just revise. This is the DNA double helix. This is the coding strand. This is the template strand. The direction of movement of RNA polymerase is from 3' end to 5' end of the template strand. Always keep in mind that the template strand is always the synthesis always occur, the transcription always occur from 3' to 5' end. Therefore, RNA polymerase is also going to move from 3' end to 5' end of the template strand. Right? And this is the hybrid region of DNA RNA. This is the polymerase and this is the transcription bubble. Now, as I told you yesterday, supercoiling takes place when the bubble comes into existing just ahead of the bubble and just posterior to the bubble. Always supercoiling takes place. We have studied in DNA replication. Like for example, if you try to open up a rope, so what does it happen when you open it up? At the point when there is a split, it tightens all the more. It supercoils. Similarly, this side where the polymerase is moving, this side is also supercoiled, which is called positively supercoiled, and the rear end shows negatively supercoiling. Positively supercoiling is along the axis of right-handed helix. Negatively supercoiling is reverse of the right-handed helix, that is in the opposite direction of right-handed helix. Now, what is this? This is the polymerase enzyme. We can very clearly see the polymerase enzyme. This helix, it is entrapped in the jaws of this polymerase enzyme. This figure is entirely different from this figure. This figure shows how the RNA polymerase and the DNA and the mRNA, how are they associated? This RNA polymerase, as we can see in this diagram, it is in the form of a jaw. And in this jaw, it is holding this DNA double strand as well as the hybrid. This will be more clear in the next diagram. Now, see, this was the jaw we were discussing in the previous slide. This is the omega subunit, which helps in association of all these four subunits. Beta subunit, and here are two alpha subunits. And this RNA polymerase is like a forceps. When you open the forceps or a jaw, and this jaw is entrapping this DNA double helix along with the hybrid mRNA DNA complex. So I hope this diagram makes the structure of RNA polymerase and the association with the complex more clearly. This was the open initiation complex, or we can say transcription bubble, which consists of polymerase, the coding strand, the template strand, and the hybrid. This is the positive supercoiling, which is present in the direction of movement of RNA polymerase, that is ahead of the transcription bubble. And this is negatively supercoiling, which is at the rear end of the transcription bubble. This part is already transcribed. This is yet to be transcribed. So positive is towards the direction which is yet to be transcribed, and negative is in the direction which was already transcribed. And this is a non-template strand, which is called as the coding strand, and this is the template strand. Again, please be careful about the direction. The direction of template strand is 3' to 5' direction, whereas the direction of mRNA is 5' to 3' end, or we should say pre-mRNA. So what is RNA polymerase? It has opened this DNA double helix so as to form the transcription bubble. And this opening is required as the helix opens. So polymerase will also move in that direction. This is the diagram of prokaryotic RNA polymerase. As we said, that there is an additional unit present in the prokaryotic enzyme. These are the two alpha, two beta, omega, and sigma. This entire is known as the holoenzyme, whereas alpha, beta, and this is known as the core enzyme. So what we were talking about, this core enzyme, 2 alpha, 2 beta, and omega, and this sigma is additional factor of prokaryotic RNA polymerase, and the entire enzyme is called as holoenzyme. Now let us read what we have already studied in the last few slides. Core enzyme is made up of 5 peptide subunits, 2 alpha, 2 beta, and 1 omega. These 4 units are organized because of the presence of this omega, helps in arrangement of these 4 subunits. And the polymerase moves from the direction of 5' to 3' direction of what? Of mRNA. Be careful, mRNA. And whereas the template, it is going to be 3' to 5'. Now this enzyme actually lacks specificity. What does it mean by? It needs an additional component so that it can recognize the promoter region in DNA double helix. How will it recognize the promoter region? How will this enzyme? Again, I repeat, this is the core enzyme which is associated, which comes into association with the help of omega, and this entire is known as the core complex. But still this enzyme is not able to identify the promoter region of the DNA double helix. So, what does it need? It needs an additional factor. In case of prokaryotes, it needs an additional factor which is known as the sigma subunit. It helps RNA polymerase to identify the promoter region of DNA double helix. And the sigma subunit plus the core enzyme, that is known as the holo enzyme. Holo is means complete. Now let us see. This is a diagram of association between the holo enzyme, the sigma factor, and the DNA double helix. And again, remember, because it is talking about a sigma factor, so it has to be a prokaryotic RNA polymerase. Now, this is the DNA double helix. There has to be some promoter region where RNA polymerase will come and attach itself. So, how will it attach itself? With the help of sigma factor. This is the sigma factor and this is the core enzyme. Entire is the holo enzyme. So this sigma factor identifies the promoter and brings this core enzyme to the promoter region. So now what happens? It is the DNA double helix, the sigma factor and the core enzyme, or we can say the entire holo enzyme. And this is the promoter site. This is the initiation site. After coming in contact, then the sigma factor, it disassociates. It does not stay with the entire process of mRNA formation because its work is done. Once the RNA polymerase is attached to the promoter region, the work of sigma factor is over and it will dissociate itself with the from the this complex of polymerase and the DNA double helix because its work is over. So what has sigma factor done? It has increased the affinity of the polymerase with the promoter site of DNA rather than the entire DNA. Again, what does it mean? That sigma factor helps in association of RNA polymerase with the promoter site of DNA, not with any other part of DNA, so that the complete enzyme gradually slides over the DNA starting from the promoter region. Now, what has the enzyme done? It has opened the two DNA strand, or we can say it has melted down the two DNA strand at the promoter region. The promoter region has the start site. Just ahead of the promoter region is the start site. And this together, what together? The sigma factor, the polymerase, the DNA, that is known as the open complex. The strand separation helps in interaction between the enzyme and the template strand. That is the purpose of opening the DNA double helix in the form of a bubble. After 10 nucleotides have been polymerized together, that means the mRNA is of the length is 10 nucleotide long. Then what happens? So after incorporation of about 10 nucleotides, there is a change in the shape of enzyme. Why is this change in shape enzyme occurs? So that it can easily move along the DNA double helix. That is the only reason for a change in shape. That is always known as a conformation. What is conformation? The change in three-dimensional shape of any molecule, that is known as a change in conformation. Conformation. Please don't confuse it with conformation. It is conformation. You have been hearing this word since long. There is a difference between the number of RNA polymerase in the prokaryotic as well as eukaryotic cells. In prokaryotic cells, only single RNA polymerase is found. Whereas in eukaryotic, starting from the simplest to the most complex one, three different kinds of RNA polymerases are found. At least three different kinds. RNA polymerase 1, 2, and 3. Whereas in prokaryotes, it's going to be single RNA polymerase. What is the function of RNA polymerase? It is utilized in the assembly of rRNAs. RNA polymerase 2, it is utilized for the formation of mRNA. RNA polymerase 3, other small RNAs, including tRNAs, 5S rRNA, it is utilized in these. And RNA polymerase 4 and 5 is used in plants, which is not utilized in animals. So for us, 1, 2, and 3. 1 for larger RNA, the subunits of larger rRNA, polymerase 2, mRNA, polymerase 3, tRNAs, and other small RNAs.